The most classic example or most known example of a retrovirus is going to be HIV. And over the next day or two, we'll probably spend a lot of time talking about how this specific virus causes the issues that it will cause. We'll start on that today a little bit. But for sure right now, I want you to have this thought that we're talking about different types of viruses or different groups of viruses that's based on what's going on with their genetics, essentially. Show you some examples. If you look at the picture here, this is of course in your book, you'll notice that this drawing or this table goes over some different viruses, and some of them are you kind of recognize. Adenovirus you might have heard of before. Herpes virus, obviously you've heard of that before. Parvo, you know what that is?
Yeah.
That affects animals. Lots of times it can affect dogs. That's a virus. Reovirus, probably you haven't dealt with that much before. But you'll notice that these different groupings of viruses are based on the type of DNA or RNA they have. Is it double-stranded DNA, or is it single-stranded, or is it double-stranded? And that could actually help you, if you were a vet or a doctor, discern, what issue the individual has. Good? Grouping things is, of course, for a benefit. Of course, there's not just these three groups. There's more groups. There's your single-stranded RNAs and your mRNAs, and your single-stranded RNAs that are used for DNA. You probably don't recognize a whole lot of these viruses that are single-stranded RNA or single-stranded RNA. But this one right here, the RNA that acts as a template for DNA, is like a retrovirus, which would be like HIV. Even some tumor viruses like leukemia are known to be retroviruses. So pretty important stuff right through there. Good. So we got this taxonomy system for the virus particles. Now let's continue on a little bit with some structure stuff. And I think one of the other things that we'll notice with the structure is that in a lot of cases, there will be a protein capsid on the outside of a viral particle that can act as a shell. So I think we want to include that in a quick little discussion. You can have a capsid that's on the outside. Of a virus particle. Of course, if it's on the outside, it's going to act as a shell. And it will also itself be built from a protein, or be built of protein, maybe we should say. Still though, one of the thoughts I think that we have is that you can't have a virus that's ever alive. All a virus can do is affect a living creature. But when we think of a virus infecting a living creature, do we think happy thoughts or sad thoughts? So usually it's like it's a detriment to have a virus come into the body. And so I think we need to talk about why a virus coming into a body, why does that cause such an issue? If a virus is non-living, then how could it do some kind of combat with a living system and shut down that living system? Like why is it a bad thought for something non-living coming in the body? And I think our discussion would have to start by talking about the requirement for a host that all virus particles have. We have said earlier that viral particles could not be classified or stated as being virus cells. Any cell is going to have an ability to reproduce, an ability to metabolize, and can be derived from all parental cells and can make their own new cells. A virus particle cannot do that. So, for sure, we could say that a host cell must be required by any viral particle. So again, any virus is going to it must have a host to do its dirty work. Let me give you a poor analogy, but at least it's an analogy. The only analogy that I could think of, or at least a decent one, would be, what if I said I knew how to install car stereos? Like I could put in subsystems. Well, that's just an idea. Like I have the knowledge up here to do that, correct? But what else do I need aside from the know-how on how to put in a subsystem? Well, I have to have a place to do this, right? I've got to have a shop with the tools. That's kind of what a virus is. A virus has got the thought. It's got the nucleic acid that could code for negativity, something bad. But just because you got the code for it, just because you know how to do it, doesn't mean you can implement it unless you have the materials to do so. That's why you need a host. Someone's garage would be like my host, right? I just think that it would not be negativity if I was putting in subwoofer systems all the time. I think it would jam. But needless to say, you have to have a host for a viral particle. It's got to have a place of business.
So you'd be the virus, the garage would be the host.
Exactly. And unless I have, if there's no garage, then what's up here is not expressed. Makes sense? Cool. I think we could also agree that a viral particle will do one of two things. They will either enter into the host cell, or they will release their nucleic acids into the host cell. So somehow the bad instructions, the bad nucleic acids that are coding for bad viral reactions are going to get inside of the host cell. Whether or not the entire particle is eaten up by the host cell or the DNA or RNA is dumped into the host cell, somehow that information is going to go inside. So going back to the example of the subsystems with my mind, I would say that either I'm going to instruct someone on how to put in the subsystems, or I myself am going to go in there and do it. But somehow, what's up here, whether I put my body in the shop or I just write down what's up here and then put that in the shop, somehow this knowledge has got to go inside of the host. Good? Okay. Now, it's not just any host. Could I walk into a garage that's working on a Mercedes-Benz and put in a subsystem? I've got to walk into a garage that actually is already built for installing subwoofer systems, right? Because that's where the correct tools are. So I could agree that it's not just any host cell. There's actually a specific host cell. And usually what happens is that when these nucleic acids are dumped into that host cell, the outcome is going to be a host cell that loses any natural abilities that it may have, any natural functions that it may have, and is likely to become a center for more creation, or for creation of more virus particles, we should say, more viral particles. So now this would actually now the story term's pretty gloom, pretty bad. This would be like I sell myself on having an idea to put in the best subwoofer systems. They let me into their garage, and I'm this evil sorcerer or something like that from a great movie, whatever, I'm a scary guy. And now that shop, that garage isn't for putting in subwoofer systems, it's for bad wizardry or whatever, I don't know. So now that shop is a bad shop because I had a bad idea. Now, instead of saying like horrible examples with subwoofer systems and garages, let's actually look at a real example. And the first thing I want us to start at is spending some time looking at a specific type of human cell. I want to look at a cell known as a helper T cell. The reason that it's a T cell is because it's made in the bone, but actually matures in the thymus, which is an organ that's up towards your neck. That's where the T designation comes from. It's where it matures at, where the cell becomes specialized. And the purpose of the cell is actually pretty simple. This cell's purpose is to recognize non-body or non-natural foreign cells and bring more immune system cells to the area to destroy the invaders. So your helper T cell is the little guy that's looking for where a battle needs to be and calls for the rest of all the immune cells to come fight the battle. So your helper T cells are essentially looking for infection sites of foreign cells. And helper T cells don't fight the battle, they just